Hello friends and welcome to my channel EG Life Science. Here in this video tutorial we are talking about a viral multiplication, a lytic cycle and a lysogeny. So please subscribe my channel and share it to your friends. The viruses multiply by a two-way lytic cycle and a lysogeny, and the number of virion particles released after one multiplication cycle from one host is called as a burst size. The lytic cycle and lysogeny are the two ways by which the viruses can replicate. Here in this lecture or in this video tutorial we are discussing about a lytic cycle of T4 bacteriophages and a lysogeny of temperate phages like lambda phage. The first, that is a lytic cycle, the phages that lyse their host after a infection are called as a virulent phages or a lytic phages. The infection of bacterium by a virulent phages result in a release of new viruses via lysis of host. The reproductive process of such phage is termed as a lytic cycle. The example of such viruses are the bacteriophage PBS1 and PBSX. These two are the example of lytic phages of a bacterium Bacillus subtilis. The phage PM2 is a lytic phage of Pseudomonas, whereas HP1 this is a lytic phage of Haemophilus influenzae. The T series bacteriophages contains 7 phages, that is T1, T2, T3, T4, T5, T6 and T7, and these bacteriophages make a number of ways for their replication, and the replication start with absorption. Before that, this is a typical structure or is the structure of a T4 bacteriophages. It has a capsid and its structure or its symmetry is a icosahedron, and in that capsid or in that head there is a enclosing of a nucleic acid, that is a double-stranded DNA molecule. This is a tail sheath, and the tail sheath is with a tail fiber and tail pins. This is structure of a T4 bacteriophages. Then these are the events which occurs in a life cycle of a bacteriophages. The step 1, 2, step number 5 and 6, these steps are of a lytic cycle, whereas the step 1, 2, 3, 4 these are of a lysogeny, and after 4th step, 5th and 6th step occurs later. Means it depends upon a natural or the natural induction. So let's start the lytic cycle. The lytic cycle start with absorption. The absorption is a first step in a lytic cycle where the bacteriophage come in contact with a susceptible host by a random collision. For absorption, the phage possesses an absorption site or an anti-receptor site, whereas the host cell possesses a receptor site. For T4 bacteriophages, the host is a E. coli bacterium. The host cell possesses the receptor site. The host cell surface components such as flagella, pili, teichoic acid, proteins, carbohydrate, lipoproteins and lipopolysaccharides serves as a receptor for a phages, and the phage components such as tail fibers, tail proteins, icosahedron spikes and knobs, this acts as a absorption site or a anti-receptor site. Each phage has its specific receptor to which it absorbs, or there is a complementation in between the absorption site and anti-receptor site. The T4 bacteriophages has tail fibers as their absorption site, and the OMPC lipopolysaccharide complex, that is outer membrane protein C lipopolysaccharide complex in a E. coli cell wall is acts as a receptor site. So there is a complementation in between these two sites. Initial absorption is weak and reversible, but it becomes irreversible when the tail pins attach to the lipopolysaccharide. And normally the tail fibers are folded around a tail. The whiskers or a collar fold tail fibers. When phage come in contact with a host, the tail fibers unfold, and for this unfolding there is a requirement of tryptophan and a cofactors like Mg ions and a calcium ions. After absorption, a next step is a penetration. It is also called as a entry, and where there is a entry of a viral genome. In case of T4 bacteriophages, the genome is a double stranded linear DNA enters into a E. coli bacterium. Once the phage attached to the E. coli bacterium, the bacteriophage, T4 bacteriophage, injects the DNA into a E. coli cytoplasm. The bacteria purges the rigid cell wall. Thus, the phages directly cannot penetrate the bacterial cells as a whole virions. Instead, they penetrate only their nucleic acid inside the cell. After penetration, there is a replication and a maturation. There is a replication and a maturation. Once the viral DNA is entered into a E. coli cytoplasm, then there is a replication and maturation. This is called as a biosynthesis. In a biosynthesis, is three steps containing a process. One is a formation of immediate early and delayed early protein. Second is a replication of phage genome, that is a double stranded DNA, and third one is that is formation of late protein. Once the DNA of virus enter into a E. coli bacterium, then the bacterium instruct to DNA for a transcription and translation into proteins. After penetration, immediately the part of phage DNA transcribed by a host RNA polymerase to form immediate early mRNA, and this immediate early mRNA get translated into two kinds of protein or the enzymes. One is a nucleases and another one is a subunit modifying enzyme. The nucleases break down the host DNA, that means the DNA of E. coli, and after breakdown of this DNA, there is a availability of nucleotides, and which are used by a replication of viral DNA. These are the precursors for replication of viral DNA. The second protein, that is an enzyme that is a SME, subunit modifying enzyme, and this enzyme specifically modify the alpha subunit of host RNA polymerase by transferring a ADP ribosyl group from a NAD, and this modified host RNA polymerase further translate or transcribe some part of viral genome to the delayed early mRNA, and that delayed early mRNA get translated into four kinds of protein or the enzymes. The first enzyme, the phage enzyme, that produce a 5-hydroxyl methyl cytosine, that is a 5-HMC, and this is a extra or a unique base in a T4 bacterial DNA, and this is produced by one enzyme. Second, that is a polymerase and ligases. These two enzymes, the polymerase, it is useful for polymerization of viral DNA at time of viral DNA replication. Ligases joins Okazaki fragments at time of viral DNA replication. Then the third enzyme, that is a glycosylation enzyme, and this enzyme adds a glucose to HMC, that is a 5-hydroxyl methyl cytosine, and this glycosylation protects a phage DNA from a host restriction endonucleases. And we know that a bacterium has a restriction endonucleases. These are the weapons to attack the foreign DNA, and to get protected from that weapons, the phage DNA get glycosylated. So hence it get secured from the action of host restriction endonucleases. And the fourth enzyme may carry out alteration of RNA polymerase once again, and here at this time there is a alteration in a sigma factor. Then the B, that is a replication of a phage genome. The phage genome or a viral genome replicate by a mode that is called as a semi discontinuous mode. It is bidirectional. Here in a replication of a phage genome, phage use a rolling circle mechanism, means the one strand get cut by a endonucleases and it get replicated by a discontinuous manner, and short fragments of a DNA are produced, and this fragment get joined by a DNA ligases, means one strand get discontinuously polymerized and one strand get polymerized continuously. So it is a semi discontinuous and it is a rolling circle mechanism. Then the third event, that is a C, formation of a late protein. Soon after a replication of a phage DNA, transcription of a late mRNAs occur, and these mRNAs translate to a different proteins. The protein includes structural proteins which are useful for a capsid production, proteins involved in a tail fiber production, protein involved in a tail plate production, and after production of such proteins, the parts of a virion get formed within a host cell cytoplasm. Next, the fourth event is assembly. In assembly, after formation of a virion component like capsid, tail pins, and replication of DNA, the complete virion particles get assembled. Number of such virion particles assembled, and the fifth step is a release. After formation of a number of virion particles inside a host cell cytoplasm, they get released from the host cell by bursting or a lysis of bacterial cell, and this bursting is because of lysozyme. The lysozyme is having it is an enzyme which acts on a peptidoglycan of a cell wall. So peptidoglycan get breaked, and from that lysis or from that pores, the new virion particles are released. So at time of release, the host cell get killed or lysed. Here in this image, the number of virion particles are formed, and they breaks or they lyse a host cell and release into a environment. The summary for a lytic cycle: absorption, attachment of phage to the host cell site surface, entry, penetration of viral DNA into a host cell, replication or a biosynthesis using viral genome to make a viral RNA and a protein, assembly, complete new virion particles are formed, lysis of host cell and release of virions. Then the next strategy used by a phage, that is a lysogeny, at time of their replication. The lysogeny the bacteriophage capable of establishing a lysogenic relationship to their host cell are called as a temperate phages. For example, a lambda phage, P1 phage, and phi A T phage. These are the example of E. coli temperate phages. P22 is a temperate phage of Salmonella typhimurium. B phage is a temperate phage of Corynebacterium diphtheria. The S1 and S2 phages are the temperate phages of Staph aureus. The temperate phages can exist a lytic cycle as well as a lysogenic relationship to their hosts, but this decision is coded by their genome. In a lysogeny, the first event or the first step is absorption. It is same to that of the lytic cycle. Host cell has a anti-receptor site and the virus has a receptor site. After absorption, there is a next step, that is a penetration. In a penetration, the viral genome get entered into the host cell cytoplasm, and there is a formation of a prophage or a provirus. Provirus or a prophage means the viral DNA get integrated into a host DNA. Here we can see how the viral DNA is integrated into a host cell DNA, and this is called as a prophage. This is called as a prophage or a provirus. And the prophage gets separated from a provirus or a host DNA by spontaneously by some natural agents, and after separation of the DNA or viral DNA, this viral DNA may continue for a lytic cycle, that is a biosynthesis, assembly and release. It is called as a spontaneous induction. Here there is a spontaneous induction, and from this point it is continue to the lytic cycle, release. This is a summary, but in a lysogeny there is a formation of a clones of infected bacteria. These are the event: absorption, entry, integration, spontaneous induction, replication, assembly and lysis. In this image, the lysogenic cycle is there, and in a lysogenic cycle, the viral DNA is integrated into a host cell chromosome, and this cell get further reproduced or divided into two daughter cells, and each daughter cell DNA has a viral DNA which is from a infection. Further, these two daughter DNA get divided and forms a number of cells. After formation of such cells or such 1000 cell or 1000 clone of infected cell, one cell from it get continue for a lytic cycle in this manner, and after lytic cycle viruses are released into a environment. Here is another schematic representation for a lytic cycle and a lysogeny. Advantages and limits of lytic cycle and a lysogeny. In a lytic cycle, replication of viruses is fast, and in a lytic cycle there is no any passing of a viral genome to the next generation of host cell. Whereas a lysogenic cycle, it is somewhat slow, and the viral genome is passed into a future generation of a host cell. So thank you for watching my video. Please subscribe my channel, press the bell icon to get a notification of my upcoming video. Like the video and share it to your friends.